Welcome to the NIAGADS Alzheimer's Genomics database, where you can interactively browse and mine data from the publicly available summary statistics deposited in NIAGADS. Datasets are standardized, harmonized, and annotated, allowing genome-wide association study results to be linked to genes and variants. The Alzheimer's Genomics database provides summary reports that allow researchers to quickly discover genetic evidence linking genomic loci to Alzheimer's disease and related dementias. In the following video series, we will show you how to use the features of the database. This video will cover navigating the homepage as well as features common to all pages on the site. Using the gene page, genome browser, variant page, and study pages can be found in subsequent videos in this series. If you need additional assistance or have suggestions for future improvements, please email genomicsdb@pennmedicine.upenn.edu. You can also find this email address in the description of each video of the series. So let's start on the homepage. This is the homepage of the database. This bar at the top here is common to all pages on the site. There's a search bar where you can enter text queries, and it'll search all the information on the site. I could enter a gene of interest, a variant of interest via a ResSNP ID or location in the genome, or even a study that has been deposited with NIAGADS that has publicly available summary statistics. There are also links to a couple of key pages on the site. First, there is a link to browse datasets. Here you can see all the NIAGADS GWAS summary statistics and click on them to explore more. Next is a link to the NIAGADS genome browser, where you can browse and inspect any of the summary statistics tracks. Finally, if you're interested to learn more about the project, click the About button, where you can learn about the project, funding sources, data sources, see commonly asked questions, and how to cite its use. Let's see the search bar in action. As you can see, when I type in ApoE, I not only get a result for the gene, but the search function will also return any dataset with ApoE in the study name, description, or sample phenotypes. Next, let's look at features common to all pages on the site. There are 3 data type report pages on the site, one for genes, one for variants, and one for studies. Each page is set up similarly, but has information pertinent to that subject. We'll cover each of these pages in depth later in the series. At the top of the page is general information about the topic. Since we are on a gene page, I see the gene name as well as some general information about the gene. Every page has an About This page at the top, which will tell you information contained within the page as well as data sources used to generate the page. Each section of the page has an Info button where you can see how to navigate the information below. You can navigate the page by scrolling up and down, or by using the navigation on the menu on the side and clicking the links. Gene and variant pages also have a button for the genome browser, which when clicked will take you to the gene or variant in the genome browser and will highlight it in yellow. You can export a report of the page or share the page. In the remaining videos in the series, we will examine how to navigate the different reports in genome browser. The next video will cover how to use the gene page.